Hi guys, quick video here about the pre-lab that you did on Friday and a wrap-up about the cladograms that you did on Friday, and then I'll make another video for the first part of the actual lab separately. So cladograms are how we show evolutionary relationships. Here is an example from that practice worksheet that I don't know how many people did because I wasn't there. But first you look at all their different traits. The idea is that if organisms have the same traits, then they probably are related, and it just depends on how closely related, how far back do you have to go to get to that common ancestor. So when everything has all cells, then they all have a common ancestor that first had cells. And of course, there's cells down here in the bottom left. But not all animals that have cells have a backbone. So at some point, a backbone evolved, and these four organisms have it. So everything after that spot has a backbone. And so right off the bat, the idea would be that if a human, a catfish, a tiger, and a frog all have a backbone, there's two possible explanations. One explanation is that each of those organisms independently evolved to get a backbone, meaning that they all got the right mutations and the changes over time to make a backbone, completely separate from everybody else for different times in the history of life. Or backbones evolved once, and we all have a common ancestor that had that backbone, and it's just been passed down and spread through natural selection and speciation. So we have this group that has all the backbones, right? Then only some of them have legs. So the frog, tiger, and human have legs. Out of the things that have legs, only the human and the tiger have hair, and of course only the human has the thumb. So we do have a common ancestor with the tiger somewhere back here. It is closer to the present than our ancestor with the frog, but the frog ancestor still exists. Right, so that's the basic idea, is that by following the lines, you can see where the branching points are, and you can figure out who's more closely related to who. So here's a practice problem that I'll move that out of the way. You could pause the video now and attempt to do this problem. But I'm going to go over the answer right now. We have these various species, and they have these traits that we don't know what they are. But we know what they have in common and what they don't have in common, and we could start to build a cladogram. So one thing to notice is that for trait number one, all of them except for Y have that trait. And so we would expect to see some sort of cladogram where Y is separate than the other groups. And so interchoice B and interchoice D are not going to fit because Y isn't something separate. It needs to be the first thing, right? Because it's the earliest thing. It doesn't have that trait that all the rest of them have. So now the question would be between A and C. And so here we see that species V and W, they have some similarities. And then species X and Z, they also have some similarities. So W and V should be similar to each other. And then X and Z should be similar to each other. And of course, that's what we see up here as answer choice A. So there's that part. Then if after you did the practice cladogram worksheet, there was some pre-lab questions for the lab. Here's another table about plants, and you can see the cladogram that I made down there that just, again, all of them had vascular tissue except for one. Then only two of them had seeds, and only one has flowers. So it makes it nice and easy when it works out that way. Again, as compared to this last one where things didn't line up so great over here on the left, all nice and perfectly. And you can see how that changes the complexity of the cladogram. So then there was the second question, comparing the GAP-DH gene in protein in humans versus other species. And these ones are nice too because you get a number, right? We just have these specific numbers right here, and we can just use those to line things up. The first question, however, is asking why is the percentage similarity in the genes always lower than the percentage similarity in the protein? And this question is asking you to remember that there's this thing called exons and introns, right? And so we're going to take our genes and then cut them apart. So the things that get cut out, they might have mutations, which would make them not as similar, only 99.6% similar here, but those pieces get cut out. So then when you look at the protein, it is 100% similarity because those mutations didn't affect the actual protein. So that's for A. Then B, it wants you to make a cladogram. And so here we have a cladogram with the worms, flies, dogs, chimps, and humans. And in this case, I don't have the little markings to show where their derived characteristics come from. I have the percentages, and I just write the percentages up next to each organism. Of course, it's important to remember that the percentages are all related to, relative to something, in this case, humans. So the human goes at the very end, and then all the other percentages are based off of that. So again, this is just to make sure that you can make cladograms and read cladograms. Once you did that, then you get to this part of the lab. So here's the basic story, that scientists find this fossil of this guy, and they want to know what kind of animal it is. And so the first thing that we would do is look at its structure. We just look at the fossil. So based on just the looks, you need to make a hypothesis. You need to put a point on the line, a dot on the line, that will show where you think the species exists, right? Does it go down here, somewhere between insects and crocodiles? Is it past the crocodiles over here? Does it go here? There's all these different places that it could go. Your job is to use the anatomy that you can see to make your guess. Then we're going to get DNA sequences and use the DNA sequences to start narrowing down and get more information, right? But that will be in a different video. So if you have any questions, make sure to email me or hit me up on Google Hangouts, and I can help you.